Hello everybody. Today we are going to discuss about the method of DNA sequencing. This is the second part of chapter 4. What do you mean by DNA sequencing? What is the sequence? What do you mean by that? DNA sequencing means reading the DNA. What do you read from the DNA? We read the sequence of base placed on this DNA. Simple as that. Okay, let me remove that. So let's say we have this template, pink colored DNA, which has the sequence of T, G, C, T, A, A, T, C. Then complementary sequence should be A, C, G, A, T, T, A, G. So how do you create this sequence? As you all know, already know, by using DNA polymerase and primer. Then we can get a complementary sequence, which is expressed in this sky blue color. Okay, so we want to know the sequence of this sky blue colored DNA sequence. Then how do you do that? How can you do it? Let's say, what if we can stop after each base? What if we can stop the polymerization after each base? Let me enlarge it. Oh, I'm sorry. Doesn't work very well here. Okay. What if we do that? What if we can stop after A, after G, after C, after T, A? Then we can get, we can read A, C, G, A. So, for example, this illustrates that. Let's say we stop the synthesis after A, then we get this small band because this is the smallest one. And let's say we stop the synthesis after C, then we get 2 base. Of course, it is attached to this blue one, the pink one. So this is the second smallest one, AC. And after that, we stop the synthesis after G. Then we stop here. We get this 3 base small band, DNA band. Then etc. Same goes on and on like that. So after each, this is the ending base. So after A we get 3. Let's say we stopped after A, then we get 3 fragments like that. And let's say we stopped after G, then we get 2 fragments like this. Can you follow me? And let's say this is thymine is the ending base, then we get how many? 1, 2. We get 2 small fragments. And then we have only 1 C, so we get 1 band here. So if you read from the bottom, A, C, G, A, T, T, A, G corresponds the sequence of this blue colored single-stranded DNA. Do you understand? This is a basic principle of sequencing DNA, and let's go on to the detail from the next slide. Let us enlarge this here. Okay, let's say we have this original sequence that looks like this, and we want to stop the synthesis after G, each G. All right. Then we get how many fragments? We get 1, 2, 3, 4, 5, 6. because we have how many? We have 6G, so we get 6 different fragments. You cannot specifically choose only one sequence because it happens randomly. So you get all these 6 different fragments in one part. All right. So how do you stop the synthesis after G? How would you do that? How would you do that? Here it says add DDGTP. What do we mean DDGTP? You know deoxy ATP, right? Deoxy is ribose missing one oxygen. What do you mean by DDGTP then? This is missing 2 oxygen because dideoxy. That's what it represents. So dideoxy GTP means the ribose ring got 2 oxygen missing. As you all know, this is the structure of ribose, right? If this ribose lose 1 oxygen, and then it becomes deoxyribose, right? And if we lose the other one here again, then it becomes dideoxyribose. Of course, here I attach it to some base here, and it is substituted with some phosphate group. Then it becomes nucleotide. But this is a concept. So dideoxy means this ribose ring lost 2 oxygen. Okay, so this dideoxy structure, if it exists in the whole batch of this solution, the reaction solution, then let's say if this dideoxy GTP comes here, then it stops here, right here. And if this dideoxy goes here, then it stops right here. The polymerization doesn't occur anymore. The termination of the chain occurs at dideoxy. Remember that. Here, this fragment, this DNA short fragment is resulting result of this dideoxy GTP incorporated into this site. The same goes on and on like that. This one goes on and this one goes here. Okay, this becomes a little messy, so we are gonna remove that. All right, do you understand the concept now? You must understand the concept. So what happens is this. Okay, this first row, this is correspond to that. Don't get confused, eh? This row becomes the column. The second row becomes this second column here. Okay? So what happens is this, like this number 1 corresponds to this band. Because why? Why? Because this is the smallest one. The smallest one migrates faster than others, so it goes down to the bottom, right? And the second one goes the second from the bottom. Here it will be placed right here. The third one goes down here, and the largest one, number 6, will be on the top, as we learned before, right? So this is the sequence. We don't know yet the sequence other than G. We have to find them too. Let me erase this and then enlarge it again. All right. So let's say we stop after C. For example, if we use DDCTP instead of G, then what's gonna happen? this synthesis will stop right here and here, 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 and here. Then how many fragments do we get? 1, 2, 3, 4, 5. We get overall 5 fragments if we use dideoxy CTP. Likewise, if we use DDATP, then what's gonna happen? DDATP. Then what's gonna happen? Alright, let me change the color. Okay. It's gonna stop here and here. Then how many fragments do we get? We get 3 fragments. Right? You understand the concept here? This is same principle. The same principle goes to other base. We didn't do the we did all with thymine. We didn't do the thymine. We will get 3 fragments here too. All right. So this is an example of sequencing through that method that was explained before in the previous slide, right? Okay, for example, we here this slide showed this dideoxy GTP. So this law here is the band from using dideoxy GTP. Here, this is the band. All right, and let me change the color again. And this is the sample from dideoxy ATP. This is a sample from dideoxy TTP, and this is from dideoxy CTP. Now you can relate the whole thing, right? So what happened here? This whole thing is from the same DNA sample. So if you read from the bottom, it starts from C, right? And the next one you cannot see clearly maybe, but I can see if you enlarge the screen here, then you can see 2 bands overlapping here. 2 bands from C here, G, sorry. So we get 2G here, and then afterwards T comes, 2 bands of T comes here, TT, and then G, etc. So you can read whole DNA sequence through this kind of method. Okay, this is another DNA sample. This sample sequence is this GAAT. You can read from the bottom. Same principle goes on here. All right, so if you the one I explained here is a traditional method, and it's time consuming and a very tedious job to do. But nowadays, it is not being done manually. It is being done automatically. That is, I will show you that the machine automated sequencer, which is called automated sequencer, is doing all that job. It does this sequencing like that. Okay, this slide shows the principle behind the automatic sequencer just mentioned now. Before we needed 4 different batches for this sequencing experiment. Remember, we needed four different batches. And as a result, four different lanes to be able to sequence the whole DNA. Okay, but nowadays things are much better. So remember, we needed dideoxy nucleotide, 4 different dideoxy, And we add these 4 different dideoxynucleotides into one pot, but we have to label them with different colors. Okay, fluorescence label. So for example, here ATP, dideoxy ATP is labeled with a green color, and dideoxy TTP is labeled with a blue color, and DDGTP is labeled with orange color, and dideoxy TTP is labeled with pink color. So you know, you can conduct this experiment in one pot. So what happens? You don't need 4 different lanes, you just need 1 lane. This 1 lane of gel. I'm sorry, it's not straight. Then you read the whole sequence by just reading from the bottom. Of course, you know, there are different ending dideoxynucleotide, but you can distinguish them by their color. Okay, so if it's blue color, then it ended with dideoxy TTP, so it should be T, and it's pink color, so it should be C. You should be able to understand this principle. So that resulting sequencing is shown here. So you see 4 different colors. It must be a very long DNA, therefore it didn't end in 1 lane, you know. There are many lanes. So you don't read these colors. This machine is reading the color and gives you the whole sequence. But you need some lots of money to buy this type of machine, of course. It's very expensive machine. But nowadays experiment gets much easier. Just by looking at this, you can easily figure that out. All right, and here you have to take a look. You have to take a look at this. What do you see here? You see something that you haven't seen before here, Taq polymerase. Can you see that? Taq polymerase. This is a polymerase that can withstand heat. It can operate above some temperature that can kill all the other enzymes. But this enzyme was obtained from some archaea bacteria that can survive at volcano or something. I think I explained this before in chapter 1. But anyhow, so this Taq polymerase is used for PCR technology that I'm going to explain now. All right, now let me explain what the PCR technology is. PCR is the short abbreviation of polymerase chain reaction. So polymerase chain reaction is for amplification of DNA. Why do we need to amplify DNA? Why? Why? Because you need to be able to sequence the DNA. If you don't have enough amount of DNA, you cannot sequence the DNA. Therefore, first you have to amplify the DNA before sequencing. Okay? And for this PCR protocol, you need 2 oligonucleotide primers. You will see that right after. And thermostable DNA polymerase, Taq polymerase. You just saw this the slide one before. And this enzyme is isolated from Thermus aquaticus, which can survive at volcano at very high temperature. Therefore, this enzyme can operate at high temperature, at 75 centigrade. Usually, other polymerases cannot survive or operate at this high temperature. There's a reason for choosing this enzyme, which I will explain later. And what else do you need? You need 4 deoxynucleotides, ATP, TTP, CTP, GTP. Okay, and of course you need the raw source template DNA. And to amplify DNA, you have to polymerize the DNA as this DNA as a template. Then what do you have to do first? Can you just polymerize the DNA without any treatment to the DNA? Of course not. You have to first melt the DNA. What is the melting? I explained it before. Denaturation. You have to separate those two-stranded DNA, double strand, into single strand DNA. Then you will be able to, let me change the color here. You will be able to duplicate the DNA, right? So you need to melt the DNA first. Okay, so for that, this whole reaction mixture is at 95 centigrade. You have to raise the temperature to this high temperature, right? Then what happens next? You have to renature. Why do you have to renature? By lowering the temperature down to 55 centigrade. Why do you have to renature? Why? Because you have this primer, small primer. Let me change the color again. Okay, let's say you have this single-stranded DNA as a result of renaturation. Then you have small primer here. This small primer should attach to this single-stranded DNA. For that, you have to lower the temperature, right? Otherwise, they cannot attach it to each other, right? So that's what happens here. And then you have to polymerize. You have to attach other nucleotides here, deoxynucleotides, let's say CGT, etc., you know. Then for that, what do you have to do? You have to give that reaction mixture to the optimal condition for that Taq polymerase, which is the temperature of 75 centigrade. So you set that temperature, raise a little bit from 55 to 75. Then this polymerization reaction start taking place. Sorry, the reaction is opposite. Okay, this reaction. Then DNA synthesis will take place and goes on and on. Okay, to help you understand better, let's watch a video animation. Actually, this is not an animation. I can explain this whole thing, but watching the video is much better for you, your understanding, I guess. Okay, hold on a second.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, now you understood PCR clearly, I'm sure. And let's say we have a protein that we know the sequence of amino acids, but you are sure that that we have that DNA that encoding that certain protein, but you don't know the sequence of the DNA. Then what, in this case, what should you do? If you don't know the sequence of the DNA, you cannot know the sequence of the primer. You first have to have primer at hand. Otherwise, you cannot start the PCR experiment, right? So you have to first produce primers, but you only know that the protein sequence starting from histidine here, let's say. This is an example. Okay, so you have histidine, glycine, and valine. Let me change the color of the pen again. Okay, so how do you deduce the DNA sequence? Here, to be exact, it's not DNA sequence, it's a primer sequence. How do you deduce the primer sequence from this amino acid sequence? Well, it's not as straightforward, you know. Here, you know, histidine has redundant codons, as you learned before. We have 2 codons, and glycine, for glycine we have 2 codons here too. For valine we have more than 2, we have 4. So the combination of all these codons, we get hold on here, we get 2 4 multiply by all 2 multiply by 2 multiply by 4, then what, how many combinations do we get? 16. So here it is, we have 16 combinations, which means we have 16 different possible primer sequences here. So are you going to try all? Yeah, you have no other choice. You know, you have the DNA, that DNA here, that correspond to one of these 16 primers. Okay, this primer here, hold on, this primer should be one of these 16. So you have to try number 1. You have to try number 1. It doesn't work. Okay, you have to try number 2. It doesn't work. You know, you have to try on, and then eventually you will find one specific, let's say this is a true primer, okay? This will be exactly complementary to this DNA of our interest. All right? So there's no other way, you have to try. This is called degenerate DNA primer. Degenerate. Because you get lots of possible primers. But thankfully, as we know that the amino acid sequence, this is possible. Otherwise, you cannot try anything. All right? Okay. And this is another story, inverse PCR. This is another also very interesting and important technology. using PCR. Okay, let's say you have known sequence. You cannot know the whole sequence. You always start from some known sequence, just like the one before we explained with the degenerate primer. Okay, so you have known sequence here, and of course there are always some restriction enzyme sites. These are the recognition sites for restriction enzyme. And you don't know these two sites here. You don't know the sequence of these two sites. And you want to know, right? Then what would you do? You only know that this middle sequence. Then this biologist, molecular biologist, they came up with a very interesting idea of doing this experiment. You first make two primers that are corresponding to this side, and the other one can attach to that side. So it starts from here. The PCR, as you saw from the video and as I explained, the PCR amplify the sequence between these two primers. Always you amplify this, okay, in large quantity. So I'm sorry, I skipped this explanation here. This recognition site, for that you apply that enzyme, and then they become sticky ends. They attach here, so it becomes like a vector, it becomes a circular DNA. That is a key point. Okay, and then afterwards, as I explained just now, you apply these two primers and then amplify the DNA between these two primers, and then the resultant PCR products are like that. Many of these DNA multiple sequences. Then what would you do next? What would you do? Now you can sequence the whole DNA here. Okay, now you can apply the primers, let's say here and here, and then you can sequence the DNA. Then you will be able to know the sequence of these two sides of question mark before. Now it is not question mark, it's exclamation mark, right? You will understand, you will know that sequence of this. Okay, so here as it is written, inverse PCR, this is called inverse PCR technology, is a trick used when sequence information is known only on one side of the target region, only one side. Then if you know only one side, you will be able to know the other side using this technology. It is a very interesting and important technology. Okay, now I'm going to explain briefly about RFPD technology. I won't spare too much of time on these technologies because they're not that easy to understand, but RFPD is quite close, very similar to RFLP that you learned before. Remember RFLP? What was the objective of that technology? They have very similar objectives. RFPD and RFLP, they all aim to understand the genetic relatedness of two different organisms. Okay? So you can judge whether these two organisms or individuals whatsoever, whether they have genetic information very close to each other or far away from each other. You can judge from this experiment using this technology. Okay. RAPD is an abbreviation of randomly amplified polymorphic DNA, and you can understand the whole thing, but if I just point to the summary, you just apply some primers. Okay, let's say you have this A and B individual. Okay, A has this DNA sequence, and these primers are attached to these sites here. Let me erase that. Hold on. Okay, and let's say B has a little different sequence than A, so maybe for B the primers may attach to maybe here or maybe here. Okay, so what's gonna happen if you do the electrophoresis? Of course, before that you have to cut the DNA with restriction enzymes, right? The same restriction enzymes. After that, okay, this is the first organism. You get these two bands. Let me tell you one thing. If the distance between two primers are too far, then PCR cannot be successful. All right? So this much distance is okay. So you get two PCR products here. And remember that we need some large quantity of DNA to be able to see the bands. Okay? So these two bands are from these two PCR products, right? And let's say this is A and in the A organism, and B organism might be here number one. Okay, so what do we see here? The band, one band corresponds to this band. They are in the same position, but the other one is a little bit different, right? This must be heavier than that. So this primer must be a little far away from each other, farther than the one with A. Okay, so this means they have certain genetic closeness. They are close in genetic information in this region, but not here. So relatively, A and B are quite close. Of course, here, this one, if you compare this first organism and number two, they are the same. They have the same genetic information. It must be, for example, this first organism must be mother, and the number two organism must be daughter or the son. Okay, they are like almost a family. And maybe number one must be some relatives, or like number three is very different from number one, right? Not number one, first organism, right? You see the band pattern is totally different, right? That means they are very far in genetic information. Okay? Okay, you know, this from these slides you can see how many different ways you can manipulate the DNA using PCR technology. That's why I emphasized many times that PCR technology is very important. You can combine any DNA. Okay, this region must be the same. The sequence must be the same, otherwise they cannot attach it to each other. Okay, they should be complementary to each other. But otherwise, you know, you can link all these three different sequence into one complete sequence. If you have two primers at the end, at two ends, and other tools for PCR, then you can make this new linked holistic DNA sequence. Okay, I'm not go down any further here. It's harder for you to understand. Okay, that is generation of insertions or deletion by PCR. You can generate the insertion I showed you here. Deletion I didn't explain any further, but you can delete the DNA by PCR. Okay, this is very important. Direct mutagenesis. Directed mutagenesis. In the textbook, it is written as direct mutagenesis. You can call it that way too, but it is more common to call it as directed mutagenesis. So mutagenic for this directed mutagenesis, you need mutagenic primer. This is a key point of this technology. And on the other side, you have to have a normal primer that are totally complementary to the gene to be mutated. You are going to mutate this gene by one base site. Sometimes mutation can be good. Mutants, after mutation you get mutant organisms. They can give you a lot of information about some information from certain genes. So to test those things, sometimes you need to mutate the gene to see what kind of phenotypes you obtain from those mutated genes. All right. So here, you can mutate in many ways, but here it does only one site. Here, this is the original sequence. Should look like the C and G, okay? But this mutagenic primer has T instead of C. You replace C with T. And after first running, you get this sequence. Of course, G and T, they are not complementary, so it should look like that. You will see a bubble here. But still, other sites are complementary, so they stick together. They stick together here, but you see small bubble, okay? And after PCR, let me change the color again. Okay, for PCR, you have to separate that, and primers come here again, and you keep running the PCR, and as a result, you will get from this bottom template, you will get this double-stranded PCR product, right? Then what do you get? Normally, T will the Taq polymerase will bring C instead of G onto this T site. Then you will get this mutation site, different from the original one, right? So finally we got mutated PCR product, and you incorporate this mutated DNA into certain vector and see the resulting phenotypes of that gene, encoding protein. Then you will understand that how this mutation and how this gene direct that information into the protein of our interest. Okay, let me briefly go through this slide. Usually Taq polymerase tend to leave A overhangs. It doesn't end with a blunt end. It adds one extra adenine nucleotide here, so it leaves A overhangs. So if you want to incorporate this into a vector, the vector should have T overhangs here. You can make it, Actually, it is manufactured too, and they can, they become like sticky ends. They easily stick together, and then you will be able to obtain this complete cloned plasmid, which is called TA cloning vector. TA cloning vector. Okay. And you can do some other tricks to make some interesting PCR products. Let's say you want to link these two sequence, number 1 and number 2, but they belong to different DNA sequence here. So how would you do that? You can overlap the primer here. You can overlap the primer, and then as a result, you will get one complete PCR product linked with sequence 1 and sequence 2. And you have already seen this, right? After DNA splicing, you get only exon linked to each other mRNA. The DNA splicing remove all the introns, right? Remember? So only exons are left. And then from this mRNA with reverse transcriptase enzyme, and you get cDNA, complementary DNA from this mRNA. Okay, and using PCR, you can obtain so many copies of complementary DNA. That's how you get cDNA of large quantity. Okay, and this is another thing that that is used in laboratory to obtain some sequence with restriction enzyme site at the end. This is very useful actually. So let's say you want to obtain some target sequence with some enzyme restriction enzyme site at the end. This must be recognition site. Then what's gonna happen? You can easily insert this resultant PCR product. It must be looking something like that, right? This says extra overhang here, and this is again extra overhang. These are all enzyme sites, restriction enzyme site. And let's say you have a vector. And then you can insert into this vector easily without any further manipulation. Alright, so in class you are going to present the actual example of PCR applications. You can find the information through internet and books, some literature, whatsoever. You can try many other ways, and also give a presentation on directed mutagenesis using PCR. You just give some actual examples. Well, it can be clinical examples and other examples. Okay, good luck with your preparation, and see you in the next class.